So we'll start with our topic, that is toxicology. In this, first we will complete some small topics like animal toxins, then fungal, bacterial, and then we'll cover metals, non-metal, and your pesticides. So this is the syllabus as per your ICAR. Animal toxin, plant, fungal, some bacterial toxins, metal, non-metal, and pesticides. And starting with the animal toxins. Starting with animal toxins. So in 2017, they have asked something about your honeybees. And in case of toxicology, the most important MCQ always you find what is the toxic principle. So in case of honeybee, they have asked one MCQ about the toxic principle present in the honeybee, and that was your melittin. This was the melittin. So this toxic principle, melittin, this is injected, or you can say it is by the help of a sting. This molecule is injected into the animal body. And this melittin, there are some other molecules also like serotonin, histamines. All these molecules, they produce hypersensitivity reaction. Of bite, sting at this point, then there may be some local reactions, or if the biting is more than enough, then there may be some systemic reactions, systemic allergy. This systemic allergy, we call this anaphylaxis. And for the anaphylactic types of allergy, the drug of choice is your.
Hello, sir. We cannot see your audio or video, sir.
Hello, sir. This acid is your cytotoxic. It kills the cells. There is one more, we call it fire ant. And the molecules or the allergic protein it carries that are derived from its
Hello?
Hello,
hello
sir.
solenopsis. Hello.
Sir, please restart this topic. So you were not audible.
Now am I audible? Hello.
Yes, sir. Yes, sir.
So Okay, I will start again. So,
Sir, we were left on honeybee.
okay. So, we have said take it, and in case of honeybee, there are many molecules like your histamine, 5-HT, but the more important is your mality. This is mality. So all these molecules that may reaction or systemic reactions. In case of local reactions, we can apply cold we can apply some cold therapy to reduce the swelling. But in case of systemic reactions, these allergic reactions they are called anaphylaxis. There are two manifestations in anaphylactic shock. One is your bronchoconstriction. You are unable to breathe properly because of bronchoconstriction. Why this bronchoconstriction? That is because of your histamine molecule. Okay. Then there is vasodilation. That is also because of histamine. So the drug of choice is your epinephrine, or we call it adrenaline. This molecule, epinephrine, act on the receptors and it causes bronchodilation. And second, it also causes vasoconstriction. So both the problems are solved. So this is your honeybee having histamine, serotonin, melatonin, allergic molecules that may give rise to local reactions or systemic. If these are systemic, we call them anaphylaxis and the drug of choice is epinephrine. Please mute your mics. I think someone is unmuted. So the second animal was our spiders. That is your
So,
black, hello, black widow
can
spider.
you start it, please?
So we missed ant, I think.
Okay, I don't know why this Bluetooth is disconnecting again and then reconnect. There are two types of ant. One is your formica. That toxic principle is your formic acid. And second is your fire ant. We call it Solenopsis, and the molecule it carries is the SOLI 124 allergic proteins. And these proteins, in case of coughs, they may cause corneal ulcers. They may cause keratitis. So all the pathology is related with the eyes of the calf. So this is related with your ant. Formica, formic acid, and this formic acid in nature, this molecule is cytotoxic. And fire ant, that is Solenopsis, carries Sole 1 to 4 allergic proteins, and in case of cough, it causes corneal ulcer. So these small toxic principles or molecules, they are asked in the exam. The next your species is the spider, and the name is your. There are two spider. One is black and one is brown. The black is more important. That is called black widow spider. The toxic principle is alpha laterotoxin, and this is neurotoxic, and that is related with interference of your neurotransmission. So let's start with this black widow spider mechanism of action. This molecule act at the neuromuscular junction. Suppose this is the nerve, this is your nerve filled with the acetylcholine. There are 100 molecules. There are 100 molecule of acetylcholine. And this is your muscle. This is your muscle. Alpha laterotoxin. It acts at the presynaptic area. Which area? The vesicle. It stimulates the vesicle so that all the acetylcholine 100 molecules depleted. Now, after its action, our presynaptic neuron vesicle becomes empty. So this is the mechanism related with alpha lactotoxin. It means the molecule causes depletion of neurotransmitter acetylcholine. All the molecules of acetylcholine they are replaced. The toxin enters, it displaces all the molecules so that these molecules they act at the muscles, they stimulate this, and at the end, when there is no molecule available, we call it flaccid paralysis. Flaccid paralysis means muscle get paralyzed because there is no availability of acetylcholine molecule. So the toxin is neurotoxic. It causes depletion of acetylcholine. All the molecules get depleted and the vesicles become empty. So if no vesicles are there and this is your muscle and this is nerve and these are your receptors, if nothing is present, then these muscles become paralyzed because there is no stimulation for the muscles to contraction, and that paralysis we call flaccid paralysis. So this mechanism of your related with black widow spider. And the second brown spider that is vasculotoxic. These are the blood vessels. Whenever this is the endothelium, suppose there is injury to the endothelium, there is one toxin and it causes injury to the endothelium. Then what start at that point is the formation of thrombus to heal that gap. So this brown spider it causes multiple injury at the multiple places, multiple thrombi formation. So this causes injury to your endothelial cell. So the brown spider toxin is toxic for the endothelial cells. If you damage the endothelial cells, which process start? Coagulation process start. Platelet comes, they deposit, then fibrin, formation of clots. So multiple clots are formed, and these multiple clots, if they are formed in a narrow lumen capillary, then they will block that capillary. This is something related with your brown spider, which is toxic to the endothelial cell. And that may lead to formation of these thrombi within the small capillaries. Next animal toxin is related with your scorpion. Similar to spiders, this molecule is also neurotoxic. This molecule is also neurotoxic. The mechanism is related with your sodium channel or with your potassium channels. Two mechanisms are related. One with the sodium channels and one with the potassium channels. So what is related with the sodium channels? That is your open. They keep the sodium channel open. It means more and more sodium will enter. More the sodium enter, more depolarization. If you continuously depolarize a membrane at the end of the day, the membrane becomes persistently depolarized and it will be inhibited. Second is not to close. First, we open the sodium channel, and second, we block potassium channel. Second, we block the potassium channel. In this way, the positive ion as the potassium move from inside to out, that will not move. That may lead to accumulation of positive charges within the cell. So in both the cases, the scorpion having neurotoxin, one component open the sodium channel that may cause entry of lot of positive charges. But in another case, the second part of this toxin that may block the potassium channel, that will not allow the potassium to move out, that may also lead to accumulation of positive charges. So in both the cases, one thing is common, that is accumulation of positive charges within the cell, and that is known as depolarization. The mode of depolarization is different, but the end result is same. Both the cells filled with the positive charges more depolarize and becomes at the end inhibited. So one part of toxin open the sodium channel, other part will block the potassium channel. And in both the cases, we are having lot of positive charges inside the cell that may depolarize our cell. So this is your scorpion toxin. Then ticks, they produce toxin. There are two ticks. Suppose one is derma center and second is your hard tick exudate. In this case of tick, we have sodium, again sodium channel. Sodium channel. In this case, they block the sodium channel. If you block the sodium channel, it means you are blocking the action potential propagation. In a nerve cell, this is the action potential that move from one part to another. If you block it at this point, so we block action potential. So the derma center take neurotoxin, they block sodium channel, they block action potential. Then what happens with your exudate? That is your hard tick. This toxin inhibited the release of the acetylcholine from this point to this point. In case of exudate, presynaptic neuron prevent the release of acetylcholine to the postsynaptic tissue. So in both the cases, neurotransmission is inhibited. If this is at the nerve, action potential stop propagating, and in this case, it block the exocytosis or release of your acetylcholine. Next animals are amphibians. There is one toad, we call that bufo, and the toxic principle. The toxic principle is derived from its name only, that is your bufogenin. That is your bufogenin. So this is very, very lipid soluble. If this is the bufo and this is your dog, and if the dog eats this bufo, then in the oral cavity at the level of mucous membrane, this lipid-soluble molecule bufogenin will enter through the oral cavity of the dog into the body, and then the bufogenin molecule will behave or act like a cardiac glycoside. It will act like a cardiac glycoside. So we call this type of poisons that they are having their action similar to cardiac glycoside. So what is that action? Suppose this bufogenin entered, then this is your cardiac cell. This is your cardiac cell. All the cells have one sodium potassium pump and one is sodium calcium. Sodium calcium. Sodium and calcium. So normally sodium enters and potassium exit. This is the function, and if you have a molecule bufogenin, that molecule behave like a cardiac glycoside. It comes and block this channel, sodium potassium ATPase. So to bring that sodium inside, this ATPase is no more functioning. So we have something sodium calcium pump. This sodium calcium bring the calcium more inside. Because of this calcium, there will be excess contraction of your heart. So it increases the calcium, and all the manifestations like your tachycardia, contraction of heart, force of contraction, all these things rises because of the calcium. So this is how your bufotode toxic principle act by increasing the calcium level inside the cardiac cell. And if these are excessively stimulated, that may lead to fibrillations and that may cause death of the animal. So this is how the bufogenin inhibit the sodium potassium ATPase. More calcium enter in, lead to contraction of heart, fibrillations, death of the animal. These are your cardiac glycoside. So next are snakes. Snakes may be poisonous or snakes may be non-poisonous. So this may be poisonous or non-poisonous. So first, the difference between these two, and uh, all the differences, all the differences they are derived from this non word, that is your no. It means everything is absent. No fangs. Fangs are absent. No heads, and in case of this, no pits and no flat tail, and there is non-complete scales on the belly. So all the features of the non-poisonous snakes start with the no fangs, no heads, no pits, no flat tail, that is hoods, and no pits, no flat tail, and no complete scales on the belly. It means the poisonous snakes, they have fangs, they have hood, they have pits, they have complete scales on their ventral portion or their belly. In case of in case of poisonous and non-poisonous, suppose this is the ventral part of the snake in both the cases. Scales are present, but these are not to the entire diameter of the ventral part of, but in this case they are present throughout their belly. So ventral surface scales are there because these are reptiles, they have scales, but these scales are not present throughout, they are present only this much part. So this is one difference. Next difference is bite mark. In case of non-poisonous snake, these bite marks are semicircular. Semicircular means you can draw them like a C, one like this. So we will have semicircular mark. Why semicircular mark? Because they do not have fangs. So no fangs mean no mark. So they have these small but several teeth that form semicircular because fangs are absent. So this is the difference between poisonous and non-poisonous snakes. Then, uh, with the classification, we have poisonous and we have non-poisonous. Then from this, we have viper type and cobra type, and their that family is called colubrid and viper. Viper. The family which contains COL, that is your cobra family, and this O will help you to remember they are oviparous. They are oviparous. Viper family start with the V, they are viviparous. They are viviparous. So they lay the larvae or animal directly give birth to young ones, but here we find egg. So in this family we have vipers, we have viper, but this Colubridae family, which is having oviparous, they are further classified into only two parts. One is hydro, that is related with the water, and second is your elapid. So hydro, PHIDAE. These are called sea snakes. These are called sea snakes because there is lot of water in the sea or in the name of the reptiles. So hydro means water, water means sea, that is your sea snake. Elapids, from the name itself, they carry La. They carry that La means they are present on the land. This family contain cobra, your great, and other. And this carry sea snake, that is your water type. Viviparidae, they contain viviparous, that is your vipers. Then what is the difference between these two types of snakes? One is your cobra family and second is your viper family. Cobra and your viper family. The first difference, these are neurotoxic, and viper, as the V tell you, they are vasculotoxic. Toxicity vessels, that is with the blood. So they are vasculotoxic, they are neurotoxic. The first difference. Then the second is related with the second is related with the eyes. These are the eyes of the cobra snake, and if you look at the name of the cobra, there is circular thing in the name, and that circular thing help to remind you that they have circular pupil. Their pupil is circular. But in case of viper snakes, this is the eye, and this V will help you. Like if you draw the V like this, it means the pupil is not circular, it is like a slit. So these are the two main difference: cobra family, neurotoxic family, and viper families. They have slit-like pupil and they have circular pupil. One more thing or one more difference in these two, scales are present. This is eye and uh, this is the nose. This is the nose. These are the scales 1, 2, and the 3, 4, and 5. 3, 4, and 5. The third, 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 3, 4, and 5. third scale touch eyes and this nasal fold. So this is one more important point with the morphology that cobra snakes, they have neurotoxin, they are vascular V for V, they have V for V, that is slit like pupil, they are having circular pupil, and the third scale touches at this side eyes and at this side nose. So the third scale will touch the nose and the eyes. Then coming to their content or mechanism of action. One more difference, one more difference between cobra and viper is mode of injection. How do they inject the venom? So they instill the venom just like a drop by drop. But in case of viper, again V for viper, they inject they inject the venom. They inject the venom and they inject like a needle. So these were some 3-4 differences between your cobra family and viper family. So most of the things of vipers they are start with the V, that is V-shaped injection or needle. They are viviparous, they have V-shaped pupil, and one more with the V, they have very long fangs. Their fangs are very long, and they are vasculotoxic. So starting with the cobra families, the main MCQs which they ask in the exam. So cobra family is neurotoxic family. These neurotoxins are of two types. One acting before the synapse and one is acting after the synapse. After the synapse. So before acting we call them presynaptic neurotoxin and this we call after means post synaptic neurotoxin. So this B will help us in remembering the whole mechanism of the neurotoxin. Before, first is before. This is the neuron and this is the muscle, nerve muscle junction. It means this neurotoxin is acting at this site, presynaptic nerve. And at presynaptic nerve, if I draw, this is your, suppose this is your cobra venom and it releases two things. One is your neurotoxin and one is acetylcholine esterase. These are the receptors present at the muscle neuromuscular junctions. This neurotoxin will bind with the receptor and block the receptor. So all the receptors are blocked so that when acetylcholine comes, it will not be able to bind. It means neurotransmitter antagonizes acetylcholine. And the second thing, the venom also contain acetylcholine esterase. If acetylcholine is present at the synapse, then this will destroy all the acetylcholine. So in this way, muscles remain unstimulated and that may lead to paralysis. This is the mechanism of neurotoxin. Those neurotoxins which act presynaptically. They act presynaptically. Uh, one, not presynaptically. This is something. 1 second. This is postsynaptic, not pre. This is your pre. In case of postsynaptic, because all the receptors they are present at the postsynaptic membrane, and the neurotoxin is going to block these, and the another molecule will destroy the acetylcholine, so that there will no stimulation and it will lead to paralysis. So all the activities they are occurring at the postsynaptic membrane. So we call this postsynaptic neurotoxic activity. So post means after. Then one is presynaptic, in something which is before synapses. This is muscle, this is nerve. So at this level, it block all the term from the B, that is before synapses. It will block acetylcholine release. It will block the acetylcholine release. And the next B is your molecule beta bungarotoxin. B for beta bungarotoxin. So the presynaptic is before the synapses, and second is it blocks the release of acetylcholine, and the third is example is B for beta bungarotoxin. And the same thing if it occur after the synapses, after the synapse, then A for alpha Bg bungarotoxin. So there are two MCQ that beta bungarotoxin act presynaptically before the synapse, and alpha bungarotoxin act after the synapse, that is postsynaptically. So the whole story of this mechanism is related with two molecules. One is alpha, one is beta. So alpha bungarotoxin and we have beta bungarotoxin. Alpha will act after the synapse, that is post synaptically, and beta will act before the synapse, that is presynaptically. Both the cases we have neurotoxin and both will lead to same result, that is your paralysis. So this was the most important mechanism related with the neurotoxicity of cobra family snakes or your bungarotoxin alpha and beta. Then fish. This is very small, that we call tetrodotoxin. Means tetra means four, odonto means teeth. The fish having four teeth and it is producing one toxin, that is tetrodotoxin. But in reality, this toxin is not produced by the fish, but this toxin is produced by one bacteria which is present in the fish, and this bacteria produce the tetrodotoxin, or also called sexitoxin. So this bacteria
What's
is
that
your
bacteria?
pseudomonas. This is your pseudomonas species. So inside the fish, there is one bacteria that produces the toxin, sexitoxin or tetrodotoxin, and that is your neurotoxin. That is your neurotoxin. And this neurotoxin, it acts at the level of sodium channel. Sodium channel. If you block the sodium channel, there will not be propagation of action potential. So this is how it blocks the sodium channel and stop the action potential. But the important question is that the bacteria is present inside the body of fish and it is producing this neurotoxin. So why this neurotoxin is not able to paralyze the fish? Why this neurotoxin block our host sodium channel, not of the fish? The reason is the sodium channel of the fish is having some kind of mutation. Because of that mutation, this toxin unable to bind or block your sodium channel. So because of mutation in the sodium channel, it will not able to bind at the sodium channel. So the mechanism is block the channel, stop the action potential that is present inside the fish, related with the Pseudomonas species, also known as Pseudo-Alteromonas. And this toxin is present in the fish. If I draw the fish edible organ, that is liver. So the tetrodotoxin is present in the liver, maximum amount. It is also present in the small intestine and other, but maximum amount in the liver, the edible part. So in case of Japan and Korea, they eat this fish after the removal of the liver because the toxin is removed. So this is the delicious fish in that locality. So the main important MCQ is tetrodotoxin, or also known as sexitoxin, produced by the bacteria, and it will block the sodium channel. Then the last animal toxin is the ciguatoxin. One fish is there, ciguatera. In the name itself, the toxin is this. And this is also a neurotoxin. The mechanism of this toxin is it will open the sodium channel so that more and more sodium will enter, depolarize the cell, and finally inhibit the cell. But the more important point, in one of the exam, they have asked one MCQ with this toxin, that was reversal of temperature sensation. It means hot and cold sensations. These sensations are reversed in case of toxicity of ciguatoxins. hot and cold sensations because it is a neurotoxin and it causes the reversal of the hot and cold sensations. This is the most important clinical sign. So after animal toxins, we'll cover fungal toxins. Fungal toxins. Fungal toxins, they may destroy your liver that are hepatotoxic or they may destroy your kidney like ochratoxin are for renal or they may related with the reproductive system. That is your zearalenone Z toxin. So these toxins may be related with the liver, your kidney, and estrogenic, that is F2 toxin, or they may directly kill the cell, that is cytotoxic, or they may damage your nervous system. So these are the five classes of fungal toxin: hepato, nephrotoxic, one with estrogen related with the reproductive system of the pig. We call it F2, and the most susceptible is your pig. And the last one is your CNS, neurotoxic. So starting with the liver. Aflatoxins. There are 4 to 5 MCQs with first aflatoxins. If we divide this name A for aspergillus, aspergillus, and this FLA is your flavus, as the name derived. So the aflatoxin, they are the hepatotoxic from the name itself, aspergillus, and the species is flavus. So there are many B1, G1, B2, Z2, but the most toxic in ICAR 2018, they have asked which is the most toxic aflatoxin. That is your B1. And which species? That is your duck, most susceptible. These aflatoxins, these molecules are lipid soluble molecules. They can easily cross the membranes and enter the organ. So there are many metabolites of this aflatoxin. Out of those, most reactive, or you can say the most active metabolite, aflatoxin metabolite is epoxide. This is the MCQ. That which metabolite is the most toxic or most active metabolite. So aflatoxin epoxide. The full name is 8,9 aflatoxin 8,9 epoxide. This is responsible for liver damage. The mechanism of action of this aflatoxin is similar to your anti-cancer drug, alkylating ones. This is the DNA and this is the guanine, the position number N7. So the aflatoxin epoxide, it will come and bind at the N7 site. Again, there will be cross-linking, less mRNA, less proteins, and that may lead to less synthesis of enzymes. And these less number of enzymes produces all the mechanisms. If defective enzymes, in case of liver, in case of liver, enzymes are defective, there may be deposition of fat, fatty liver. That is your fatty liver because of these enzymes. So the main target of aflatoxin epoxide was N7 of guanine in case of DNA molecule. And the things which are related with the liver is your fatty liver. And the second thing is necrosis, centrilobular necrosis. Necrosis. If I bring this one lobule of the liver outside, then around the center there will be necrosis. We called it centrilobular necrosis in the lobule of liver around the center. And the second is the fatty degeneration, we call accumulation of fat. So this fatty accumulation and centrilobular necrosis, and because of these fatty things, the liver increases in its size and large liver. So this was your aflatoxin. Second, which damages the liver, that is just remember the name rubra-toxin B. Rubra-toxin metabolite B. This is also hepatotoxic. Then with the renal toxins, we have okra toxin. They damage your kidney. And the MCQ is which part of kidney is damaged most commonly? That is your PCT. So okra toxins, they mainly damage PCT, that is proximal tubule of the kidney. And because of this, all the functions of kidney, they are affected. Non-steroidal estrogenic compound, that is your F2 toxin or zearalenone. The metabolites are alpha and beta. Metabolites are alpha and beta. Alpha present in the pig and beta present in the cattle. In case of pig, the disease is known as vulvovaginitis syndrome. Vulvovaginitis. This estrogenic compound, it will act at the receptor of the estrogen. The molecule will act as, and it will decrease FSH. It will decrease or FSH secretion, and FSH is responsible for the follicle maturation. So there will not be any follicle maturation. So the estrogenic receptor stimulator F2 toxin that is of alpha form, beta form, alpha in pig, beta in cattle, and this causes vulvovaginitis in case of pig. Then there is one more toxin, T2 toxin. They simply ask about the mechanism of the T2 toxin. So T2 toxin will bind with the 60S subunit of the ribosome and interfere with the protein synthesis inside the cell. One more MCQ with T2 toxin is the radiomimetic action. It means just like the radiation, suppose this is the radiation kill the cell, similar to that T2 toxin directly kill the cell. So this direct cytotoxic action is called as radiomimetic action. Then another is ergot poisoning that may be acute and that may be chronic. And this chronic C4 chronic is common in case of cattle, and this lead to dry gangrene. And this was the MCQ in 2017 that ergot fungus causes dry gangrene in case of cattle. In which form? Chronic form. So in case of cattle, this dry gangrene or chronic form is, and this gangrene is of the extremities. That is very important point. Dry gangrene of the extremities. In case of chronic ergotism, animal cattle. And the acute form is not present in cattle. It is in sheep or dog and horses. But in case of cattle, that is important. Dry gangrene of extremities. Then there are two important toxin, bacterial toxin. One is botulinum toxin and another is your tetanus toxin. Suppose this portion is the part of spinal cord. From the spinal cord, what comes out is your motor nerve. Inside this, vesicle is there and acetylcholine is filled. There are some molecules, they help in fusion of this acetylcholine vesicle and then release of the acetylcholine at synapse. These molecules, they are known as synaptobravin or syntaxin and SNPs. So these proteins, they help in fusion and then release occur. But the botulism, B for botulinum toxin, it will do B for block the acetylcholine release. It block the acetylcholine release. In case of tetanus, again, this is spinal cord. At the level at the level of spinal cord, if we bring the spinal cord outside, then there are some small inhibitory neurons at the motor neuron. and this is your muscle. Normally, from the higher level of CNS, action comes and it stimulates the nerve that will stimulate the muscle. But that action cannot remain at that place forever. It means you have to stop this action. Once the action potential comes, the muscle contracts, then action stopped. To stop that action, we have inhibitory neurons in the spinal cord. And these inhibitory neurons, they secrete inhibitory molecule, that is your glycine in spinal cord and GABA in the CNS. So in case of tetanus toxin, in case of botulinum, in both the cases, there will be muscle paralysis. But the mechanism is different. In botulinum, it blocked the release of the acetylcholine from these vesicles to this area. If acetylcholine is blocked, not released, nothing is available here. So muscle get paralyzed, and this paralysis is known as your flaccid paralysis. Flaccid. So how the botulinum toxin block the release? It start cutting this molecule. If you remove or cut all these synaptobravin, syntaxin, SNP molecules, then vesicle will not be able to fuse and it will not be able to release the acetylcholine. So this point is your mechanism of action, that these molecules are cut down, cleaved by the botulinum toxin, and this occurs. Flaccid. In case of tetanus, the action site is your inhibitory neurotransmitter. This tetanus toxin come and act at the inhibitory neuron. Similar to this, suppose I bring this small neuron outside. This is your inhibitory neuron, and this is the vesicle filled with the glycine, and these are the synaptobravin. This like this molecule cut the synaptobravin, and it will not allow the interneuron to release glycine on the next. It means the inhibitory control over the motor neuron is totally gone. Once this inhibitory control is gone, then there is nothing to control this neuron. It will keep on firing, and it will stimulate the muscle. Muscle will contract continuously, undergo tetany, and that is called your spastic paralysis. Spastic paralysis. So this is very important mechanism. If you have any doubt related with botulinum and tetanus toxin, because in many exams they have asked the same. Both the cases, paralysis is there, but in this case, acetylcholine, the excitatory neurotransmitter release is inhibited. In this case, glycine release is inhibited, so that the negative control is not there and the neuron is continuously stimulating the muscle. And because of that, it undergoes tetany, spastic paralysis. So these are two molecules. Next is poisonous plants. Your poisonous plants. First is your cyanogenetic plant. Cyanogenetic plant. Cyanogenetic plants mean those plants, they have cyanide molecules, but that is not active, and The examples are almond and your sorghum. The molecule is known as amygdalin, and here you call it dhurin. They will release cyanide molecule. So they are known as cyanogenetic plant. So bitter almond carry the molecule amygdalin and the sorghum carry the molecule thurin. And these molecules they liberate cyanide. So the species which is more susceptible, they are your ruminant. So ruminants are more susceptible. Then in case of normal body, to metabolize this cyanide, we have one enzyme that is your rhodanase. It will metabolize the cyanide. But in case of toxicity, if the concentration is high enough that your enzyme is saturated, then it will lead to the toxicity. So normally, the enzyme which metabolize is your rhodanase. The mechanism of action of this cyanide molecule is at the level of electron transport chain. There are four complexes in ATS: 1, 2, 3, and 4. The fourth complex take the oxygen and there will be production of water. Some ATPs are formed. At the level of this complex, there is conversion of iron plus 3 into plus 2 form. So the cytochrome carry Fe plus 3 which keep on converting Fe plus 3 and Fe plus 2 forms. The main MCQ with this molecule is the cyanide. It combined with which form of the ion? So cyanide combined with the Fe plus 3. Cyanide combined with the Fe plus 3 so that this Fe3 plus 3 will not be able to convert into plus 2 form and the cycle stop. Then how to remember this? Cyanide. This is the structure of cyanide. There are 3, 1, 2 and 3. 1, 2 and 3. So this is Fe plus 3. It bind with the Fe plus 3 and block the ATS, no ATP synthesis, and the animal dies. So this is important, Fe plus 3. The antidote or the treatment is your sodium nitrite and thiosulfate. Normally, cyanide combine with the cytochrome and form a complex, cytochrome complex. We have hemoglobin molecule. Along with this, we have sodium nitrite. Both the things combine and they form met hemoglobin. They combine with each other and this cytochrome is removed, cytochrome is released, and this cyanide is combined with this molecule. It will become cyan, this complete molecule met hemoglobin. Then we add our second drug, sodium thiosulfate. Sodium thiosulfate. This sodium thiosulfate, this thiosulfate give sulfur molecules which combine with the cyanide and it will become HCl and it will be excreted in the urine and hemoglobin is recovered. So this is the treatment where we use the drug nitrite and sodium thiosulfate to remove the drug through the urine. Then there is one pea plant, Lathyrus plant. There is one pea plant that may cause two types of osteo and neuro. This affects the musculoskeletal system, that is bones, and the toxic principle in this is important, BAPN. And in case of neurotoxicity or neuroletharism, the molecule is ODAP. That is your uh, oxalate diaminopropionic acid. So this molecule, they say this is structural analog of the glutamate. Glutamate. So in the brain, it behave or act like a glutamate. That is excitatory neurotransmitter. It causes the excitation of the brain and it also causes the degeneration of the neurons. And all the clinical signs like paralysis of legs, bladders, and other part of the body that they are related with this neuroletharism. But the osteoletharism this molecule is related with the collagen synthesis inhibition. If you look at the arteries, in the wall of the arteries, collagen is present, and this molecule BAPN it decreases collagen synthesis. So the wall of the artery become thin and it will rupture. So this pathogenesis with this toxin, osteoletharism. Means collagen cross-linking, collagen synthesis is not there because it inhibit the collagen synthesis or cross-linking that may lead to the weakening of the wall. And the wall become weak, then the vessels may rupture. So not only the blood vessels, in case of bones, we have collagen. We have collagen. If this toxic principle cause the less synthesis of collagen or less cross-linking of the collagen, bone becomes weak. And all the features, they are related with the lameness. So with the blood vessels and with the bone, collagen is related. And this osteoletharism toxin BAPN act on collagen cross-linking. And it may rupture the vessel or make the bone weak and bending of bone, weakening of bone or lameness in case of animal. Then the next plant is bracken fern. There are three toxic principles. Bracken fern. The first will destroy your vitamin B1, thiaminase B1. It will destroy the thiaminase, destroy the B1. So there will be deficiency of this vitamin. Second is the aplastic anemia factor. Anemia factor. So aplastic anemia is the term related with your bone marrow. This term is related with the bone marrow. It means this molecule will suppress the activity of bone marrow. So bone marrow suppression, aplastic anemia factor. And the name of this factor is PTA quiloside. So that is related with bone marrow suppression and aplastic anemia. One is thyminase, destroy the B1, and the third factor which is having resemblance, this is called quercetin, and quercetin is co-carcinogen, co-carcinogen with some papilloma viruses, and it causes cancer. So these are the three factors in the plant black and fun. One destroy your B1, cause the deficiency of B1. One will suppress your bone marrow, that is aplastic anemia factor, PTA quiloside, and the third is your quercetin, which is acting as a carcinogenic with papilloma viruses. So it stimulates these viruses activity, and there will be papillomatous growth, formation of the papilloma. Next we have some plants oxalate. The molecule of oxalate is like this. Two negative charges. So this is related with your calcium. In the blood, the oxalate will chelate the calcium. And because of deficiency of calcium, all the calcium dependent activities are inhibited. Like hypocalcemia will decrease the heart contractions, muscles, nerve, so it will lead to neuromuscular weakness. If this calcium is decreased in the blood, then coagulation will be defected. And in case of bone growth, in case of milk production, so all these things. One more important with this oxalate is in case of sheep, this was the MCQ in one exam. In case of sheep, it interfere with cellulose digestion. So one mechanism was related with the calcium, and second is the cellulose digestion. Means if the animal is feeding with oxalate diet, that may cause ruminal dysfunctions. And in case of sheep, the pH of rumen alter, pH alter. And because of pH alteration, the bacteria which digest the cellulose, that will hamper. So there is cellulose digestion impairment in case of sheep, in case of oxalate poisoning. So these are two most important mechanisms. Then there is one with diagnosis of this oxalate poisoning. Diagnosis of oxalate poisoning. So calcium oxalate crystals, they are deposited in your kidney. So calcium oxalate crystals in the kidney and rumen epithelium. Epithelium. So these are two places where calcium oxalate crystals are diagnosed in rumen epithelia and the kidney. This confirms the diagnosis of the oxalate poisoning. Then treatment, because the oxalate causes hypocalcemia, the treatment will be your calcium salt. Calcium salt is your treatment. Then there are some plant which causes photosensitization, like your lantana camera. Photosensitization. This is your plant, and the green part of the plant carry chlorophyll. This is the chlorophyll. And the pigment present is in higher concentration in case of this green part, leaf, stem. So when the animal take this green portion, they eat the green portion, then this pigment will enter and it will circulate in the blood of the animal. Then it will reach at the level of liver. Liver will convert the molecule into non-toxic form. And out of those breakdown product, there is one product, phyloerythrin. Phyloerythrin. This is the molecule or the breakdown product of chlorophyll, which is responsible for the photosensitization. So in the GIT, suppose the chlorophyll is also digested in phyloerythrin, then this phyloerythrin will be removed by liver, and if it is not removed, this molecule will circulate in the blood. And when it comes in contact with the sunlight, this is the skin, and this in the skin, this is phyloerythrin. When it comes in contact with the sunlight, this is called photosensitization. So in case of photosensitization, there will be dermatitis. There will be dermatitis. So there are many plant which causes the dermatitis. So if I summarize this one, photosensitization, this is the plant and animal eaten the plant. Inside the GIT, there are bacteria, there are bacteria, and these bacteria convert the chlorophyll into phyloerythrin. It enter liver and then through the bile or feces goes out. Because of excess, it will enter into the circulation, phyloerythrin, and then from circulation it comes at the level of skin. When sunlight falls on the skin in non-pigmented areas, that may lead to dermatitis. This is the whole story of photosensitization. And remember the name of the plants, lantana plant. It causes photosensitization. Then one more plant is your datura plant, and the active principle is your atropine. And atropine is the parasympatholitic drug. So all the activities from the secretion point of views, either it is tear or it is saliva, all the activities are stopped because of atropine poisoning. So the antidote is your physostigmine, is your physostigmine. So the question is, why physostigmine is antidote? Why not neostigmine? Why not neostigmine? So answer to this question, this is the CNS having brain and spinal cord. And from this CNS, what comes out? These peripheral nerves. Suppose it goes to the heart. Some peripheral nerves, they goes to the eyes. So the atropine is non-selective drug. It is non-selective drug. It will act at the CNS and also act as the peripheral organs. So we want a such antidote for atropine which also enter the brain and act at the periphery. So if you use neostigmine, your neostigmine will not be able to enter inside your brain. So the manifestations which are related with the peripheral part, that can be reversed with neostigmine. But these CNS manifestation, they are not reversed by neostigmine. So we want a drug like physostigmine, and this physo will tell you this is soluble in lipid. So the lipid soluble drug physostigmine, it can enter CNS and reverse atropine, or it will act at the periphery, also reverse atropine. So this is the reason why physostigmine is the antidote, why not neostigmine? Because of its lipid soluble component. Then next is oleander. The important point is act like a cardiac glycoside. Cardiac glycoside means this is the cell sodium potassium ATPase, sodium and potassium ATPase. Then we have calcium and sodium channel. This will block the sodium potassium ATPase, what enter inside more and more calcium. That may cause contraction of your heart. So oleanders act like a cardiovascular, just like your bufosine. So this oleander mainly causes cardiotoxicity. Sometimes they ask about this, that the mechanism is similar to glycoside and toxicity related with the cardiac component. Then we have ricin molecule that is obtained from the castor bean, ricin. It is related with the ribosome. It means it is related with the protein synthesis inhibition. Protein synthesis inhibition. 60S subunit is inhibited by the ricin molecule. There is one more term, heparin component. This also acts as the ribosome, but the site of action is 26S. So these are two important points with the ricin. They inhibit 60S subunit and this inhibits the 26S subunit. And overall protein synthesis is inhibited. There is one more plant that is your related with cotton seed, and the molecule is gossypol. Two MCQ related with this. One is the corpus luteum Cl lysis. It causes the lysis of corpus luteum. And second is it is polyphenol. It, the molecule is phenolic in nature, and second it causes, in case of cattle, lysis of the corpus luteum. So luteolytic action on the ovary. So these were some plants and their toxic principles and with their mechanisms. Now there are metals and some non-metals. The first metal is your arsenic. The forms of arsenic as per the toxicity is arsine gas, then +3, then +5, then organic. So this is the order where the molecules are toxic. This gas is most toxic. Then out of the inorganic form, +3 form is more toxic than the +5 form. So this arsenic in old days, it was used as homicidal drug, homicide, to kill the human being, and also as used as malicious poisoning, means to kill the like animal like your cattle. In case of arsenic molecule interact with the sulfhydryl groups. Wherever it finds sulfhydryl groups of proteins, enzymes, it will interact and make a complex. Suppose these sulfhydryl group are present in your glycolysis cycle, your Krebs cycle, ETS, TCA, it will inhibit or block all those enzymes. Then the energy pathway, energy production will be decreased. So it will inhibit the glycolysis or TCA cycle. So the first mechanism is the inhibition of sulfhydryl group. Second is related with the capillary. Arsenic damages the capillary, then the fluid comes out that may decrease your blood pressure called hypotension, and hypotension may lead to shock and death. If these capillaries are present in the renal system or renal capillaries are involved, then because of this fluid loss, less blood will reach to the kidney, less GFR, less filtration, and then less excretion means oligouria, finally anuria, and acidosis. There will be necrosis of the tubular part of nephron. So these are two mechanisms, capillary damage or sulfhydryl group with the arsenic +3 form, plus arsenic +5. If you look at the periodic table, nitrogen, phosphorus, this, these are the members. So there are two members. One is your arsenic, one is your phosphorus. Both belong to the same periodic table group. In case of bone, we have phosphorus. If this phosphorus is replaced by the arsenic, this is replaced by the arsenic. And similarly, in case of mitochondria, there is ATS cycle, ATP production is there, and in case of phosphorus, arsenic enter, that may lead to less ATP, less energy. So this is substitution of the phosphorus with arsenic. That is from your periodic table because they are chemically belong to same group. Then organic arsenic. Organic means it will enter your brain. It is lipid soluble. So in case of pig, the molecule can cause blindness. This was the MCQ. Blindness caused by the organic, and second, because of muscle weakness, it causes dog sitting posture in case of pig. So this is the organic arsenic because the neurons damaged by this. Vitamin B1 thiamine that is essential for the health of the neurons. Its activities interfere by organic arsenic. Plus, this is the neuron and this is myelination. It causes demyelination. So all these lead to muscle weakness and dog sitting posture. Second is your blindness. And the last form of arsenic is the gaseous form, ASH3. In ICAR 2017, they have asked arsine gas is hemolytic in nature. Hemolytic. It means this gas break the RBC and cause hemolysis. And the route is your pulmonary. Inhalation route, gaseous molecule oxidize the RBC membrane, damage it, cause hemolysis, and this is not related with your sulfhydryl group. So if we give the antidote dimer-caprol or BAL, it will not be effective because there is no inhibition of SH group. It simply causes hemolysis and pulmonary edema because it is inhaled. In 2020, one MCQ was on clinical sites, arsenic clinical sites, and that were watery, rice watery diarrhea. Because the arsenic causes damage to the capillary of GIT and irritant action, is causes diarrhea, projectile diarrhea or rice watery diarrhea. And because of this diarrhea, dehydration, hypotension, all things occur. And chronic toxicity in case of arsenic is rare because it is rapidly excreted by the kidney. So we will not find chronic toxicity. Then diagnosis, as your arsenic bind with the SH group, and this SH group mostly found in your keratin protein. Keratin is found in nails, hoof, your skin, your hairs. So it will found at these places. So for diagnosis, we can take this structure like hairs, and plus it also damages liver more than kidney. So in case of liver, we will find fatty degeneration. So hairs, livers, and the last thing is the blood. Blood is not a reliable method of detection of arsenic. So we do not use blood as a sample. So we can use these samples or liver or kidney. And the antidote is dimercaprol or your British anti-Lewisite, BAL or dimercaprol. This will chelate the arsenic. The mechanism is chelation and remove the arsenic. One more molecule, that is lipoic acid, 2019 ICAR. Which of the following can be used as antidote for arsenic? Lipoic acid. Lipoic acid and British anti-Lewisite. Both the molecules, they chelate the arsenic and make a complex and remove it from the body. Then the next metal is the mercury. There are three forms of the mercury. One is elemental, which is present in your thermometers. One is inorganic and one is organic. This organic form is responsible for the disease Minamata disease. Inorganic form is GIT corrosive. It will cause diarrhea. It will cause diarrhea. And elemental, which is present as vaporized form, so this will be related with the pneumonia. If the vapors are inhaled, that may cause bronchitis and pneumonia. So ruminant species are more susceptible to the mercury. So this organic will be lipid soluble, it will cross the brain and cause the nervous manifestation like visual cortex, interfere and cerebellum. So all these are nervous system form and the mechanism is similar to arsenic, that is having affinity with your sulfhydryl group. In case of mercury, there is one post-mortem finding that liver size reduces. Normally, in toxicities, size of the liver increases, but in case of mercury poisoning, size of the liver reduces. Mercury also bind with the SH group, so the treatment is same, BAL or we call dimer-caprol. So this is the mercury molecule complex. It bind with this and remove or make the SH group free of the mercury. The next metal is lead. Lead causes neurotoxicity, nephrotoxicity, immunotoxicity, and many other, like your hematopoietic system. So this molecule in the synthesis of hemoglobin, there are some enzymes which are inhibited by your lead. Remember two enzymes, one is delta ALAD dehydratase and second enzyme ferrochelatase, also called hemoglobin synthetase. So these are the two major enzymes which are inhibited by the lead molecule in the synthesis of hemoglobin. One is alpha ALAD, that is aminolevulinic acid dehydratase, and second is the heme or hemoglobin synthetase. So if we block this enzyme, then hemoglobin synthesis will be reduced. Then in case of dog with the lead, there is one concept, that is basophilic stippling. If this is the RBC, inside the RBC we'll find multiple blue color dots. This is basophilic stippling. These are the dots, and these dots are nothing but your ribonucleic acid accumulations. So basophilic stippling is found in dogs in case of lead poisoning because of inhibition of some enzymes like nucleotidase enzyme. Because nucleotidase is related with the nucleic acids, so these are the aggregation of ribonucleic acid. Then the treatment, we use calcium disodium EDTA as an antidote for the lead. Why the calcium is added with this? Because in this combination we are giving EDTA. If the drug of choice or antidote is Na2, calcium Na2 EDTA. So this will chelate your calcium. So we give calcium with the molecule so that there will not be any kind of hypocalcemia. So this is the reason why calcium is combined with the drug. Then with the copper, next is copper. The most susceptible species is your sheep. Sheep is the most susceptible. There will be accumulation of copper in the liver, and this is the most susceptible. Second, in case of dog, there is autosomal recessive mutation. In case of a breed known as Bedlington Terrier, Bedlington Terrier, that dog breed also susceptible to the copper poisoning. Then copper inversely to molybdenum and sulfate. If there is excess molybdenum, then there will be deficiency of copper. So we can use this inverse relationship in case of toxicity. If the copper is more, then if we give molybdenum and sulfate as a treatment, so we can decrease copper toxicity. So molybdenum and sulfur are given in the treatment of copper poisoning. In case of copper
Sir,
poisoning
can you
in
repeat copper once more?
okay. In case of copper, sheep is the most susceptible species. In case of copper, there is one dog breed, Bedlington Terrier. In this breed, there is recessive autosomal mutation that may cause retention of copper in the liver. So the mechanism of copper is related with RBC membrane oxidation, that is hemolysis. So there is a term hemolytic crisis. More the RBC will damage, it will lead to the hemolysis and loss of the RBC. So the mechanism in case of copper in acute toxicity, there is one sign we call greenish diarrhea, and this was the ICAR 2017 MCQ. That greenish tinge diarrhea seen in which toxicity? It is seen in copper toxicity. And the second with the copper toxicity is your gunmetal kidney. What is the concept of this gunmetal kidney? It causes, these are the RBC filled with hemoglobin. If hemolytic or oxidation of the membrane occur, what comes out? Hemoglobin. This free hemoglobin, it enters the kidney, nephrons, and start damaging the glomerulus. It may cause glomerulonephritis, damage the tubules. So in this way, the kidney will become dark colored because these glomerular capillaries, they are filled with the hemoglobin. They are blocked by the hemoglobin. They become darkened. We call it gunmetal or gray colored kidney. So these are the dark black kidneys because of this hemoglobin deposition. So with postmortem finding, there are two important things. One is pulpy enlarged spleen because the RBC, they enter the spleen and size of the spleen increases. So pulpy and large spleen. And second thing is the kidney, glomerulonephritis and gunmetal kidney. And third is the greenish colored ingesta or greenish colored diarrhea. These three things are important about kidney: pulpy spleen, gunmetal kidney, greenish tinge, diarrhea. The antidote first is D-penicillamine. Penicillamine. Second is with your copper inverse relationship molybdenum and sulfur. So if we give molybdenum and sulfate, they decrease the copper absorption and they decrease the toxicity of copper. And also we have vitamin C. In case of dog, it decreases absorption of copper. Absorption of copper. So these are the three drugs we use in the treatment of copper poisoning. Then similar to copper, we have molybdenum. As we have seen, copper inversely to molybdenum. So when there is toxicity of copper, it means in that area there is when there is toxicity of molybdenum, it means there is deficiency of copper. And the species which is most susceptible to molybdenum is cattle. In case of copper, it was sheep. So all the factors which decreases the copper, they may lead to molybdenum excess. So copper deficiency may causes the molybdenum excess. And in case of treatment, if molybdenum is excess, then we can utilize this relationship of copper. If we increase the copper, then we can decrease the toxicity of molybdenum. So this is the copper sulfate as an antidote. Then there are some other antidotes than this. Okay. Then there are three important non-metals. One is your fluoride. As we know, fluoride is related with the teeth
And
and
metal,
what?
selenium.
Selenium is non-metal, no? Selenium is non-metallic. In case of metal, there is thallium and cadmium. From that portion, they just ask the antidotes only. So if you remember, that is enough for that. Yes. Selenium is non-metal. It is not metal. So in case of fluoride, there is bone and teeth. Fluoride carry one negative charge. In case of bone, hydroxyl ion is there if there is deposition of some calcium hydroxyapatite structure. Then the fluoride replace the OH and that deposit itself at that place. Make the bone weak and there is bending of the bone. Similarly, there is teeth development, odontoblast cells are there, and these cells may be damaged by your fluoride. That may lead to the mottling of teeth. So, odontoblast or these ameloblast cells they may be damaged by the fluoride, and that may lead to the mottling of teeth. But in case of both, this is the fluoride ion which replaces the hydroxyl. And because of that replacement, instead of calcification, more and more fluoride is deposited. That may lead to the highest concentration of fluoride in the bone. So that clinical signs with this will be your lameness, and with the teeth there will be mottling of or brown color, brown yellowish color of the teeth. And as this is the negative charge, so after ingestion, once it enter the GI tract, fluoride, it will find HCl in the stomach. So in the stomach, with the HCl, it reacts and form more toxic compound HF, that is highly irritant corrosive molecule that may damage or irritate the mucosa, or it may cause all the GI tract ulcerations and inflammations. So this is the molecule. Then fluoride is an anion molecule. So second mechanism, it may combine with the cations of the body like calcium, like magnesium, and decrease their concentration. If calcium is decreased, nerve conduction, muscle conduction, all the conductions are impaired. And one more is fluoride inhibit acetylcholine esterase enzyme. If this enzyme is inhibited, then acetylcholine will not be breakdown. It means ACH concentration will be high. And because of high acetylcholine, there will be more cholinergic symptoms. So these were three mechanisms related with the fluoride. One is with your gastric acid. It will give corrosive action. Second with the cations, and third with the ACH. And the toxicity is mainly with the teeth and the bone. So the treatment, because fluoride ion decreases calcium, so the treatment will be calcium salts. Then there is one selenium. There are some plants. There are some plants which accumulate the selenium, and they cause the when the animal eat these plants, then there will be toxicity. And these plants may be obligate accumulator, or they may be facultative accumulator. So these plants, they require this selenium and they accumulate inside their green leaves or stem. So in case of animal body, the role of this selenium is related with the selenium is mainly related with the growth of hoof, growth of hoof, growth of hair, nails, these structures. If there is toxicity, then hoof structure will be destroyed, cracking of hoof that may lead to the lameness. So this selenium molecule replaces the sulfur. The sulfur containing amino acid are methionine and cysteine. In case of these two, what is present is your sulfur. Selenium replaces these, and this molecule, these amino acids, they are present in hair, they are present in hoof. So if we replace sulfur with the selenium, then structure of hair or hoof will be altered, and that may lead to weakening of hairs or cracking of hooves. So there are three types of selenium toxicities: acute, subacute, and chronic. In case of acute toxicity, this selenium molecule enter the animal body with the help of ingestion of the plant. If large number of plants are eaten by the animal, then selenium toxicity will occur, and this selenium will irritate the GIT, and there will be diarrhea. There will be diarrhea, watery diarrhea. If excess plant is eaten, then excess selenium will enter, that may lead to irritation or a corrosive action that may lead to watery diarrhea. In case of subacute toxicity, we call this blind stagger. Blind stagger. It means the animal becomes blind. The vision is impaired in case of subacute toxicity. So initially the animal vision is good, but as the toxicity increases, there is impairment of the vision. And at the final stage, the animal become blind. And there will be death of the animal because of respiratory failure. And the chronic one is called alkali disease. And there is cracking of the hoof. Cracking of the hoof, that is, there is lameness in case of cattle, in case of horse, and the structure of the hoof altered, it may become large size hoof. Structure also deformed. There may be cracks. It becomes rocker bottom uh, hoof, means the size increases. So all these are related with the chronic toxicity. In case of birds, the chronic toxicity, the embryos or the hatchability of the egg is compromised. Means the chickens which are born, they are born without beak and with some small feathers or absence of feathers. So in case of selenium, there is no specific antidote, but there are some contraindications. We cannot use, in case of selenium, the contraindication is dimer caprol. Why the dimer caprol or British anti-leucide we cannot use in this case? Because it will further increase the kidney damage. Because of kidney damage. And second thing is the vitamin E. Suppose there is selenium toxicity and we administer vitamin E. Vitamin E has a synergistic action with the selenium, so the toxicity will further increase. And as a part of treatment, we can increase the protein diet. If we increase the protein diet, more methionine will come, more cysteine will come. If the selenium replaces all these, then excess of methionine they will reduce the toxicity of selenium. So a high protein diet is very important because it will provide cysteine, methionine, sulfur containing amino acids. Then we have phosphorus. There are red phosphorus, white or yellow. This red is inert, but the white or yellow is the reactive, highly reactive or highly dangerous, highly toxic. So the toxicity of phosphorus is mainly of the yellow phosphorus. This phosphorus molecule, this phosphorus molecule is a strong irritant. It means if the molecule enter the gastrointestinal tract, it will damage the mucosa, stomach mucosa, and that may lead to gastroenteritis and diarrhea. Second mechanism is damage, cardiotoxic damage. It also damages directly to the heart. We call it cardiotoxic effect. Phosphorus, if it enter the skin, if it enter the skin, it may cause burn at the entry point. So on dermal exposure, it will cause burn injuries. So there are two type of toxicities of phosphorus. One is acute and one is chronic. Phosphorus has two, acute and chronic. Chronic is known as PHOSS, Fossi-Jaw. And in case of acute, there is gastroenteritis. There is gastroenteritis. Means there is diarrhea, hemorrhagic diarrhea, bloody diarrhea. because the phosphorus is GI irritant. And in case of chronic toxicity, phocyjaw means there is necrosis of the jaw bones. There is necrosis of the jawbone. Necrosis in case of phocyjaw. So the tooth structures, teeth which are attached with the jaw, they become loose and there will be offensive discharge at that area and the tooth become loose and there is necrosis and they may fall out. In case of phosphorus poisoning, there is no antidote available. We have only supportive therapy. Just to increase the diet of protein and carbohydrates. This phosphorus is lipid soluble, so on dermal exposure it causes burn injury. So oil are contraindicated. Oil diet is contraindicated because in case of oily fluid, oily media, there will be more absorption. So oil or lipid diet they increase the absorption of phosphorus. So these are contraindicated. We cannot use mineral oil to just like activated charcoal we use for the absorption. So we cannot use oil to further increase the toxicity. Then we have nitrite or nitrate toxicity, NO2 and NO3 toxicity. So in case of ruminant, they have bacteria in the rumen that if NO3 is coming from the diet, they can convert that into nitrite and further into ammonia and then into protein. So in case of ruminant, they can utilize this in this form. But non-ruminant, they will not convert this pathway from nitrate to nitrite ammonia protein. So out of the two forms, which is more dangerous or if we say that cattle are more susceptible to which form? So cattle or ruminant, they are more susceptible to NO3, nitrate poisoning. They are more susceptible to the nitrate poisoning. And in case of non-ruminant, they do not have any enzymes to convert this NO3 to NO2, so they are less susceptible to nitrite poisoning. So if we look at these two molecules, NO3, NO2, then becomes ammonia or microbial protein. So if there is excess of nitrite is given in case of non-ruminant. In case of non-ruminant means horse and pig. So there is no mechanism to convert this. It means they are less susceptible to this poisoning. But if we give direct NO2 minus, then there will be more toxicity. There will be more toxicity. So this NO2 molecule, this NO2 molecule, this 2 means it will combine with 2 hemoglobin. Combine with 2 hemoglobin and convert that into met hemoglobin. Met hemoglobin cannot carry oxygen. It is not able to carry oxygen. So there will be deficiency of oxygen tissue level. So this NO2 bind with 2 molecule of hemoglobin that may cause hemoglobin into the met hemoglobin. Normally, in our body, we have some enzymes. For example, there is hemoglobin. If it convert into met hemoglobin, so normally we have some enzymes that convert this back into hemoglobin. But in case of toxicity, these are saturated and concentration of methemoglobin will increase. And that is responsible for your brownish color of your the plasma. So this is brown color molecule, coffee color, brown color uh, blood will be there. Because in case of poisoning, all the enzymes which are normally present, they are saturated and excess of the methemoglobin will not be able to convert back into the hemoglobin. So that gives our postmortem finding that the blood is chocolate brown color. Then how to treat this nitrate toxicity? We have one drug called methylene blue. But the problem with this methylene blue is that it is oxidizing agent. Oxidizing agent. Our hemoglobin from Fe plus 2 then Fe plus 3. It is already oxidized. We want a drug that reduce the Fe plus 3 into Fe plus 2. But this drug is itself oxidizing. So we convert this drug first into reducing agent that is known as leucomethylene blue. That is known as leucomethylene blue. So we convert first this molecule into reducing agent with the help of some NADPH enzyme system. So this leukomethylene blue, now it acts as a reducing agent and it will convert Fe plus 3, that is your met hemoglobin, into your normal hemoglobin. So that is a tricky point with the methylene blue. Itself, this is oxidizing agent, but our hemoglobin is already oxidized. So we want a reducing agent to take it back to the hemoglobin. So first we convert into reducing agent, then it will reduce the methemoglobin back into the normal hemoglobin. So this is the treatment of choice, methylene blue. Or we have one more option. Instead of converting this into this, we can use directly vitamin C. Vitamin C is a reducing agent. So it will reduce the methemoglobin back into the hemoglobin. Or there is one supportive treatment, that is your cold water. Cold water. Inside the rumen, we have enzymes which convert NO3 into NO2 minus. This cold water, it will suppress the activity of enzymes so that the nitrate will not be converted into the NO2 toxic form. So the use of cold water is to suppress, inhibit the activity of enzymes so that less NO2 will be there. So similar to nitrate, the same mechanism followed by your ClO3 minus and NO3 minus. Both are same. Both convert hemoglobin into met hemoglobin. In chlorate also, same treatment, methylene blue is the treatment to convert this back. So chlorate and nitrate, chocolate brown colored blood, and both the cases there is hemoglobin to methemoglobin formation, and to convert it back we use methylene blue. Or directly, if you want to use direct reducing agent, then the choice is vitamin C. So there are two examples which are related with the dark chocolate color blood, chlorate and nitrate. Then there is one non-metal, sulfur. Sulfur poisoning is not common in domestic animals. Sulfur poisoning is not very common, but still this is a GIT irritant drug. So the mechanism, two mechanism, one is GIT irritant, and second is, as we have studied, copper inversely to molybdenum and sulfur. So it will antagonizes copper. So as it is GIT irritant, so it will cause diarrhea. It will cause diarrhea. But the more important with the sulfur, In case of rumen, this sulfur lead to formation of one gas that is known as H2S gas. So the smell of H2S gas, diagnosis for the sulfur poisoning. And this smell is also known as rotten egg smell. So this smell is diagnostic for the sulfur poisoning. Only pesticide portion is remaining. So pesticide, we'll do pesticide with tomorrow's lecture, systemic pharma. Because in systemic pharmacology there are three systems are important: GIT, respiratory, and your kidney. Means your diuretic. So these are mostly, and the one is your autonomic nervous system. So these four comes into your systemic pharmacology. Other topics like your immune system and endocrine, they are not that much important. So we'll cover like GI,
Ca-
all drug, respiratory, diuretic, and autonomic, and pesticide with tomorrow. Hello, hello.
And cardio drugs and
Cardio drugs. There is one classification in cardio drugs like cardiac glycosides, which we call digitalis, that act through your sodium potassium ATPase, and then antiarrhythmic drugs. That classification 1A, 2A, 1 and 2, 3. Because cardiac system is related with your beta receptors. On the heart, we have beta 1 receptors. So that portion will be covered through your autonomic nervous system. Because in autonomic, we'll cover alpha 1, 2, 3 receptor, alpha 1, 2, beta 1, 2, 3. So at that time, all the drugs will be covered at this part only. Because heart carries beta 1 receptor. So, and one more thing is the cardiac glycoside disturbance. So that is small topic. But these are the major portion from where they have asked two questions or three MCQs. So it's 12 p.m. Uh, pesticides and systemic will cover tomorrow. Okay.
so can you share the module of the today class?
In today's class module, I need some time because there is some editing or I have to add few MCQs with that. So tomorrow's by I think 2:30 I will set before 2:30 after
Okay, sir.
modifying that one. Okay.